While the tissue involved in growth and reproduction cannot withstand the test of time, it is possible to reconstruct the development of some dinosaurs with help from the fossil record and a closer look at their modern counterparts. Starting at the most basic stage of development, we can now understand the conditions under which dinosaurs laid their eggs. By taking a closer look at the report by Deeming, we can tell that the construct of an eggshell can tell a lot about the environment from which it was laid. The most descriptive figure in this regard is the level of water vapor conductance, or how well the egg absorbs ambient gaseous water. We see how this process applies to many extinct species. Almost all birds and many geckos nest their eggs in an exposed environment where the eggshell gas conductance is relatively low. Reptiles, grubs, and megapode birds, however, bury their eggs in a substrate where humidity levels reach near saturation, where the eggshell conductance is relatively high. With these examples in extinct nature, we can infer that a high water vapor conductance equivalates to buried eggs in a humid environment, and low water vapor conductance equivalates to exposed eggs in a dry environment. The main question in this paper looks to answer whether or not dinosaurs buried their eggs in a substrate like reptiles, or left their eggs exposed like birds. The term seeking clarification here is incubation, the method of keeping eggs in a suitable environment for the growth of the embryo, and for our purposes, how did dinosaurs care for their eggs? By reviewing the structure of dinosaur eggshells and specimens, we can gain a better understanding of how eggs were incubated. In his paper, Deeming mentions his gratitude to the information provided by scientist Zelenitsky. Using a Nikon Coolpix 990 digital camera mounted on a pedographic microscope, Zelenitsky photographed 2 to 10 eggshell fragments from each specimen available. Along with the help of Adobe Photoshop and the Imaging Processing Toolkit 4.0, she obtained statistical information concerning shell pores. Deeming also used data from other sources and recalculated structural statistics found so that all data used in the experiment matched the control variables. Many variables like pore area, egg mass, and egg surface area work together to ascertain statistical information. All the variables are provided in Table 1. The equations found in Table 2 organize measured and calculated data to find new measurements. However, the most important character of the egg was the general pore size. This abbreviated version of Table 3 provides the statistical results from the equations and measurements. While naturally these results are difficult to interpret, they do lead Deeming to an implied conclusion to his query. Do dinosaurs incubate their eggs by burying them in a substrate, or resting them in an exposed nest? Through the analysis of the data and calculations obtained by Deeming and by comparing these information to the morphology and eggshell structure of extinct species, he found that many dinosaurs maintain a high water vapor conductance. In fact, dinosaurs primarily buried their eggs in a substrate. While reproductive mysteries still surround many species, Deeming agrees that by correlating his findings to potential findings, dinosaurs primarily incubated their eggs in a substrate. In another report, Zelenitsky takes a close look at egg clutches of theropods to determine the conditions under which they laid their nest eggs. Theropods, a group of carnivorous bipedal dinosaurs, includes the maniraptoran clade, which is composed of birds and all other dinosaurs closer to birds than to ornithomimids. In this study, Zelenitsky takes a close look at a specimen found from the 2 Medicine Formation of Montana. The 2 Medicine Formation is primarily composed of sandstone, limestone, and mudstone, and is dated to be a part of the Upper Cretaceous period. The specimen itself, named Montanolithus strongorium, is composed of a nest trace of 5 partial eggs. The eggshell thickness, density, width, length, position, and other microscopic details of Montanolithus was recorded and then compared with the data known of other theropod nesting traces. From this data, it was concluded that the dinosaur that most likely laid this nest was the Sixotaxa tessitipodi and Troodon. Additionally, it was concluded that the females constructed their nests in sandy, poorly vegetated areas. Based on the positions of the eggs, it was determined that females simultaneously formed 2 eggs in their oviducts at one time, and laid these on the slope of the mound, while afterwards they used the central flattened top of the mound as a resting place, as can be seen in extinct species of birds today. The fossil record is also useful in helping us determine the growth strategy and development of dinosaurs following their embryological stage. In this report, Bybee and his associates take a closer look at the growth rate of Allosaurus. The Allosaurus was one of the most common theropod dinosaurs in the late Jurassic. It was spread over a wide geographical range in North America and Portugal, where paleontologists have found more than 1,000 bones representing over 60 dinosaurs. With a big skull balanced by a heavy tail, the carnivore was very fast and an ambush hunter. The average lifespan of this dinosaur was anywhere from 22 to 28 years of life, measuring roughly at a height of 5 meters, a length of 12, and weighing around 1.4 tons. The purpose of the study was to determine at what age was the Allosaurus typically its largest, and at what time in its life was the growth rate the fastest, and if the findings of this study would correlate to other studies of theropods with similar size. For this purpose, 6 humeri, 5 ulnae, 6 femora, and 3 tibiae of fibromallear bone of Allosaurus were tested. These bones were chosen because they represent a range of ontogenic stages from juvenile to subadult Allosaurus. To conduct the growth strategy in the ontogenic limb bone scaling of the Allosaurus, scientists perform a histological analysis and image analysis. Histological analysis, or histology, is the study of the cellular organization of body tissues and organs. Used to perform this analysis, a light microscope or electron microscope is used to study the structure of both tissues and cells and the correlation between the two in their physiological function. In the histological analysis, periosteal and lines of arrested growth circumferences were measured because they can be easily used to predict body mass and are easily comparable across sections of a given bone series. The periosteum is the membrane several cell layers thick that covers most of all of every bone, while lines of arrested growth are dense lines corresponding to the growth of long bones on radiograms that demonstrate the temporary slowing or ending of longitudinal growth. Bybee and his associates found that the humera, femora, and tibiae are all circumferentially orientated, meaning that the bones grew by encircling old bone, whereas the ulnae are longitudinally oriented, meaning that the bones grew by extending out. Based on an ontogenic series of these 4 sections of fibromallear bone, the scientists estimated that the ages of the largest Allosaurus to be between 13 to 19 years. In addition, they also found that the growth curve reconstruction suggests that the maximum growth of the dinosaurs occurred at 15 years, when body mass increased at a rate of 108 kilograms per year. Finally, they found that the growth dynamics and limb scaling is comparable in theropods of roughly the same size. For example, they found the lengthened bones increase in length more slowly than isometry predicts, which is similar for tyrannosaurids. As can be seen from the diagram, the bones of the Allosaurus are its largest from 13 to 19 years of age, with a maximum growth rate of 15. In conclusion, growth strategy and ontogenic limb bone scaling of Allosaurus resemble those of comparably sized theropods. Like other theropods, their 4 major bones are highly vascularized fibromallear in texture and suggest rapid periosteal growth. Another report by Erickson and his associates follows the growth patterns and sexual maturity in non-avian dinosaurs. In this study, the fossils of Oviraptorids, a group of bird-like dinosaurs, and Deinonychosaur, a small theropod dinosaur of the Cretaceous period, were used alongside the fossils of Deinonychus antihorpus and Troodonformosus to determine whether sexual maturity was reached before or after full adult body size was reached. The fossils of each sample were found in breeding positions alongside nest traces, and their bones were used to determine maximum body size and longevity of each dinosaur. From this information, age versus body size plots were then made, and the developmental stage in which each dinosaur in the sample perished was determined. Histological and growth curve patterns were constructed based on the maturation patterns seen in living reptilian species and can be shown in this following chart. From these results, it was determined that the dinosaurs from this sample ranged from 6 to 18 years of life. Additionally, some of them reached their sexual maturity only after obtaining their full adult mass, while others did not. In conclusion, despite their many modern avian features, the life history of non-avian theropods differs substantially from those of present birds. In conclusion, although the soft tissues associated with growth and development cannot be preserved in the hard fossil record, bone and eggshell structures can be easily used to provide the missing information required to fill in the life history of dinosaurs. Additionally, taking a closer look at the dinosaur's closest extant relatives can help us find growth and development patterns that cannot be easily found through the fossil record.